If you like this video, please subscribe. Thank you.
All right, so gluconeogenesis. As the name indicates, neo is like making new. Glucose is the synthesis of a new molecule of glucose. So before I start, I want to just remind you, the best way to approach each pathway is to say, what is this for? Why do we need gluconeogenesis? If you can't answer that, then whatever details you learn won't be meaningful. You want to be able to answer it by saying, we need to make glucose when we are fasting, because certain tissues and cells depend on it, in some cases almost entirely on glucose in the bloodstream for their energy supply or energy needs. So that's the first important thing, to really get a big a picture for why we need gluconeogenesis. Second important thing I'm going to remind you of is where it takes place, meaning what tissues and what part of the cell. So that's a two-part question. So I'm going to say to you, gluconeogenesis takes place in two tissues only: liver and kidney, and the liver being the most important of the two. Second part of where is that it takes place both in the cytoplasm and mitochondria. So you have enzymes in both compartments. So the reason we want to make new glucose to resupply the bloodstream, remember, this is a glucose producing as opposed to glycolysis being a glucose consuming. Very important. Appreciate the difference. This is a pathway that will produce, make glucose. Glycolysis consumes glucose. So again, gluconeogenesis is therefore most likely to occur when you are in a fasting state. And what you want is to make glucose, to restore blood glucose, so that tissues that are entirely, like red blood cells are entirely dependent on glucose for their energy. They have no other energy producing pathway. You should have appreciated that after glycolysis. That's the only pathway that red blood cells have for energy production, is glycolysis. So they need a constant supply of glucose. So do other tissues, including the brain. The brain is not exclusively dependent on glucose, but for the most part it really is. It can only use ketone bodies, but for the most part the brain is also highly dependent on glucose. Testes, kidney, medulla are also dependent on glucose. So the one thing I want you to think about is, this is kind of an important big picture. So what is the important organ that's going to resupply the blood with glucose? The liver. It's the main organ where gluconeogenesis occurs. The kidney does a little bit, but 90% of glucose that's made and released to the bloodstream is made in the liver. So this is another important thing to kind of keep an eye on. The tissues. So here are some of the tissues that really require a constant supply of glucose. Glycogen stores can work for producing glucose as well. However, their these stores in the liver are limited, meaning there's a finite amount of glycogen. So anyway, we will cover that in the next block. So you need to make glucose. Once this amount is exhausted, you have to make glucose from other smaller molecules. And these smaller molecules are listed here. 3 types of smaller molecules you are starting with. Amino acids and the keto acids derived from those amino acids, and we'll talk about those. Glycerol can be used, but it's really a minor precursor. And importantly, both lactate and pyruvate are very important precursors for the synthesis of glucose, de novo synthesis of glucose. Okay, again, 90% of that occurs in the liver. So in many ways, I want you to think about gluconeogenesis as going backwards, the reverse of glycolysis. Let's say you start with pyruvate, you're going to end up with glucose. So you're going backwards in glycolysis. However, you have an important issue here, which is the 3 rate-limiting enzymes, pyruvate kinase, PFK1, and glucokinase in the liver are all irreversible. So for those 3 steps, you have to bypass those respective irreversible enzymes. Okay? So those are the new enzymes that are required in gluconeogenesis that will bypass these irreversible steps. The remaining 7 steps in glycolysis are reversible. So that means the same enzymes are used for those in gluconeogenesis. Okay? So you with me? Okay. So we said primary source is going to be amino acids and keto acids derived from amino acids, lactate / pyruvate, and glycerol. So basically, think about this. You're going to go from here back to make glucose. So note how most of these steps are reversible, with the double arrows, except for step 1, this one, this one. I can't see there. Let me see. Let me go to the next one. Okay, so this one, glucose to glucose 6-phosphate, that's glucokinase, hexokinase, that's irreversible. Pyruvate to PEP, that's pyruvate kinase, right there, is irreversible. And this one, fructose 1,6-bisphosphate, PFK1, is irreversible. So you have 1, 2, 3 irreversible steps. The rest are reversible. So you have to bypass these steps. Okay, I guess you didn't like what I was saying. Okay, so, all right, so let's start with pyruvate. Because this step is irreversible, you have to come up with something else. In fact, you're going to need 2 enzymes to convert pyruvate back to PEP. Okay? So this is going to require 2 enzymes, and we'll start here. So we're going to start with these 2 enzymes that are required to convert pyruvate back to PEP. So first of all, I want you to appreciate this important thing here. Pyruvate If it's produced from glycolysis, let's say, where is it? In the cytoplasm. It can actually be produced, I want to point out to you, from other sources like alanine, the amino acid, and give you pyruvate. Let's say you had some pyruvate in the cytoplasm, or lactate that went to pyruvate in the cytoplasm. Before pyruvate can undergo gluconeogenesis, and note this is the direction of gluconeogenesis, it's the reverse. You have to bring in pyruvate into the mitochondria, and there's a specific transporter there that brings it in. So let's say you bring in pyruvate into the mitochondria. That's where gluconeogenesis begins. In the mitochondria, you're going to convert pyruvate to a molecule called oxaloacetate by adding a CO2 to it, a carboxylic group, from 3 carbons to 4 carbons. This is the first enzyme here in the mitochondria called pyruvate carboxylase. Converting pyruvate to oxaloacetate. The rest of gluconeogenesis is going to be in the cytoplasm. Okay? So you have to bring that oxaloacetate out. But the mitochondria have this impermeable membrane. So before you can bring that oxaloacetate out, you have to convert it to something else. And you convert it to malate. You reduce oxaloacetate to malate. Malate can be transported. There's a specific transporter for malate that will take it out to the cytoplasm. But let's focus first on this enzyme here, the first one, pyruvate carboxylase. We've already covered that. It's the first bypass to bypass the pyruvate kinase step, okay, in glycolysis. This enzyme is present only in liver and kidney and not in muscle. That's why muscle will not have gluconeogenesis. 2 important things about this enzyme. You're going to need ATP, CO2 as for the reaction, and the coenzyme vitamin biotin. Okay? So here's CO2, here's ATP, and this is the enzyme, coenzyme biotin needed for pyruvate carboxylase. And that, again, will occur in the mitochondrial matrix, okay, to form oxaloacetate. This is a little bit too much detail, but it's just to show you, really, you don't need to know all the details, except here's biotin attached to the enzyme. And it is the carrier of that CO2 that eventually gets incorporated into the oxaloacetate. So that's how some of these coenzymes function. They play a key role in the transfer of what's being added here. In this case, it's a CO2. So all you need to remember is you need biotin for pyruvate carboxylase, you need CO2 and ATP. So you form oxaloacetate in the mitochondria. Like I said, the rest of gluconeogenesis, going up back to glucose, is going to occur in the cytoplasm. So you have to transport oxaloacetate. This is the mitochondria, if you're not sure. So you have to transport that out. You have to convert it first to malate before it can be transported. One of the most important allosteric regulators of pyruvate carboxylase, that enzyme we just covered, is acetyl-CoA. Small molecule typically produced from beta oxidation of fatty acids in the fasting state. So think about it. This high amounts of this molecule will activate pyruvate carboxylase allosterically. You should know what allosteric means. It binds to the enzyme at a separate site and activate the enzyme from the catalytic site and activate the enzyme. Okay? So once you produce oxaloacetate, we convert it to malate by malate dehydrogenase. Malate is then transported out to the cytosol, where the rest of the enzymes required for gluconeogenesis will be. So the rest of the way, it's all going to occur in the cytoplasm. So oxaloacetate, malate, once malate is in the cytoplasm, it is going to be reoxidized to oxaloacetate. Okay? In the cytoplasm. The malate conversion to be able to transport it out of the mitochondria. Once in the cytoplasm, it's reoxidized by the same enzyme, cytosolic malate dehydrogenase, gives you oxaloacetate. Oxaloacetate now is going to be converted to PEP. So this is the second part of that bypass. Really, we're trying to make pyruvate, PEP from pyruvate, so we need two enzymes in succession. Pyruvate carboxylase was the first one to produce oxaloacetate. The second one is called PEP carboxykinase. And that converts oxaloacetate to PEP in the cytoplasm. So that's oxaloacetate. PEP carboxykinase uses energy from GTP to produce phosphoenol pyruvate. Okay, so this is kind of an energy utilizing step, because this is a high energy molecule we are producing. So in order to form PEP from pyruvate, you need in succession pyruvate carboxylase, then which is in the mitochondria, then PEP carboxykinase, which is in the cytosol. After you transport malate out of the mitochondria and then reoxidize malate to oxaloacetate. Okay, so this is again the big picture. This is the first enzyme. Here's the second enzyme. Okay, this is PEP carboxykinase, and this is pyruvate carboxylase. This one is allosterically regulated, activated by acetyl-CoA. So know the location, know what they're producing. Okay? So if you produce PEP here from PEP carboxykinase, now you can go backwards in gluconeogenesis, in glycolysis, because all of these are reversible. Okay? So you can go to phosphoglycerate kinase, 3-phosphoglycerate, and so on, until you get to the irreversible step, which is the PFK1 step. Okay? So that's step number 3. You go all the way back from PEP to fructose 1,6-bisphosphate. You can't reverse that because that's an irreversible enzyme that produces that molecule, PFK1. So what you have to do is you need a different enzyme. And that enzyme is called fructose 1,6-bisphosphatase. Okay? Fructose 1,6-bisphosphatase is the reverse, if you want, reverse enzyme of PFK1. It's a separate enzyme. However, this enzyme is similar to PFK1, is allosterically regulated. So here's the enzyme. You produced fructose 1,6-bisphosphate from the first two enzymes in gluconeogenesis, went back on using the reversible steps until you get to this point. This enzyme, its function is really to take the 1-phosphate off and produce fructose 6-phosphate. But look what's inhibiting this enzyme. Fructose 2,6-bisphosphate, the same molecule that activates PFK1 inhibits this enzyme. However, under fasting conditions, glucagon is high, which activates the glucagon receptor, activates cyclic AMP formation, protein kinase A. Protein kinase A, if you remember, I'm gonna go to this slide here, it's easier to see. So these are protein kinase A will phosphorylate the bifunctional enzyme PFK2 and FBP2. That's the enzyme. PFK2 makes fructose 2,6-bisphosphate, and this one breaks it. When these guys are phosphorylated by glucagon-dependent pKa, this is protein kinase A, look what happens. FBP2 phosphatase, fructose bisphosphatase, that's what this is, FBP2, is active. So that means it can hydrolyze fructose 2,6-bisphosphate and removes it from the cytosol. So this is the key thing. Glucagon is high because glucose is low. It phosphorylates the bifunctional enzyme, making the enzyme that breaks down fructose 2,6-bisphosphate more active. So you see its amount decrease here. Do you understand this? And then at the same time, PFK2 becomes inactive when it's phosphorylated by pKa. So it can no longer produce any more of this fructose 2,6-bisphosphate. And as a result, if you remove, let me go back here, if you remove this molecule by FBP2, it's no longer inhibiting fructose 1,6-bisphosphatase, and then the reaction can go. So fructose 1,6-bisphosphatase, under these conditions, this is really what you want, is going to be in the presence of glucagon. Fructose 2,6-bisphosphate will be removed because of the activation of fructose 2,6-bisphosphatase. Okay, you remove this molecule, it no longer can inhibit fructose 1,6-bisphosphatase. That's the important bypass enzyme that you want for gluconeogenesis. Okay, are you thoroughly confused? Okay, so in order for this enzyme to work, this will probably confuse you some more, but I am gonna go here. In order for the bypass enzyme to work, you want to remove this molecule. How you remove it? By activating, by phosphorylating the bifunctional enzyme. The bifunctional enzyme FBP2 phosphatase, FBP2 phosphatase, fructose bisphosphatase 2, that means it removes the phosphate from the 2 position. Will remove this molecule from the cytoplasm. If you remove that molecule from the cytoplasm, it can no longer inhibit fructose 1,6 bisphosphatase. Okay? So the bypass enzyme is functional and can work. So in addition to fructose, so what we want to do is to remove fructose 2,6 bisphosphate, so fructose 1,6 phosphatase can work on fructose 1,6 and reverse that irreversible glycolytic step. Okay, so you remove the allosteric inhibitor by phosphorylation of the bifunctional enzyme. Phosphorylation of the bifunctional enzyme activates FBP2 phosphatase, which removes fructose 2,6 bisphosphate, and inhibits PFK2, preventing it from making any more fructose 2,6 bisphosphate. Okay? There are 2 other allosteric regulators of fructose 1,6 bisphosphatase, the bypass enzyme, and they're kind of the reverse ones for PFK1. It's inhibited by high levels of AMP and activated by high levels of ATP. Okay? These 2, ATP will inhibit PFK1 and AMP will activate PFK1. So the roles are reversed. You with me? Kind of? The point is to remove the amounts of fructose 2,6 bisphosphate. And the enzyme that's going to remove fructose 2,6 bisphosphate is FBP2, or fructose bisphosphatase 2. That enzyme gets activated by pKa-dependent phosphorylation, and therefore it can hydrolyze fructose 2,6 bisphosphate and removes it from the cytosol, thus removing the inhibition on fructose 1,6 bisphosphatase, or FBP1. Okay? That's the best way I can summarize it. So you need a little time to go over it. That's why I say you need to finish glycolysis before you go here. If you're still not sure about glycolysis, finish that and then go here. You need to understand that bifunctional enzyme that contains PFK2 and FBP2. Okay. So now, if you form fructose 6-phosphate from fructose 1,6 bisphosphate by fructose 1,6 bisphosphatase, or FBP1, you can go through phosphoglucoisomerase backwards to glucose 6-phosphate. Okay? I missed, there's not a slide there, but remember, there is a reversible step. Step 2 in glycolysis is reversible. So you have to go back from fructose 6-phosphate that was produced by fructose 1,6 bisphosphatase back to glucose 6-phosphate. And then from glucose 6-phosphate, what you need really is glucose. And that's the last bypass enzyme that you need. The last bypass enzyme is glucose 6-phosphatase. It is going to remove the phosphate from the 6th position of glucose. Because what the liver needs to make is glucose, not glucose 6-phosphate, glucose. Because it wants to release that glucose in the bloodstream. You cannot release glucose 6-phosphate. You can only release glucose. So this is a unique enzyme present in the liver and kidney, which allows both of these organs to produce glucose through gluconeogenesis. Okay? The muscle does not have that, so it cannot produce glucose through gluconeogenesis. Only the liver and the kidney, to a limited extent, has a glucose 6-phosphatase that converts glucose 6-phosphate back to glucose. So this is what the enzyme does. Takes that glucose 6-phosphate back to glucose. This enzyme is actually in the ER of the liver, and then the glucose produced is produced in the lumen of the ER and transported into the cytoplasm first by GLUT7 transporter, if you remember, and then out of the cell by GLUT2. The liver has the GLUT2 transporter that can work either way, picking up glucose or releasing glucose. In the case of gluconeogenesis, you are producing glucose in the liver, so the liver is going to release it to the bloodstream eventually. Okay? So what you want to focus on really here in gluconeogenesis are the bypass enzyme, pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase or FBP1, and glucose 6-phosphatase. And obviously the regulation of each one. So we were talking about glucose 6-phosphatase. There's an important inherited disease called von Gierk's disease, where there is a deficiency in this enzyme. So the liver is unable to produce glucose. The last step in gluconeogenesis is inhibited, and the liver cannot resupply the blood with glucose. And that causes severe hypoglycemia. A very important disease called glycogen storage disease. Type 1.
If you like this video, please subscribe. Thank you.